Hello, and thank you for joining us for today's presentation, part of our Adjuvant Cell Analysis Global Conference. I'm Susie Valdez of Labroots, and I'll be your moderator for today's event. So let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you want during this presentation. To do so, simply type them into the Ask a Question box and click Send. We will answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing this presentation, simply click on the Ask a Question box located on the far left of your screen. I now present today's presenter, Dr. Kirk McManus, an associate professor in the Department of Biochemistry and Medical Genetics at the University of Manitoba and senior scientist at the Research Institute in Oncology and Hematology at the CancerCare Manitoba, Canada. For complete biography of Dr. McManus, please visit the Biography tab at the top of your screen. Dr. McManus, welcome. You may now begin your presentation.
So my research team is interested in identifying the molecular origins of cancer. The second interest that we have is exploiting that information to develop novel therapeutic targets that selectively exploit those defects. Now, to enable this research, we've had to develop and utilize a myriad of different types of quantitative imaging microscopy approaches. So really today, what I'm hoping to do is present some of our findings, but of course, some of those specific approaches. We are very much interested in genome instability and its relationship with cancer. In fact, many of you will know that up to 95% of all cancers exhibit some form of genome instability. And there are many different forms. There's three that are listed here: microsatellite instability, CpG island methylated phenotype, and what we are interested in, chromosome instability. Chromosome instability is defined as an increase in the rate at which whole chromosomes or large chromosomal fragments are gained or lost. And in the context of a population of cells, this is very much a driver of cell-to-cell heterogeneity, and it's often that heterogeneity that we are assessing within those quantitative imaging microscopy approaches. CIN is arguably best understood in colorectal cancer context, where up to 85% of all colorectal cancers exhibit CIN. CIN generally comes in two flavor formats. Shown on the top left is numerical CIN, where basically we can silence one of our favorite CIN genes and convert a karyotypically stable normal diploid cell into something that has massive increases in chromosome numbers. The other type is structural CIN, or S-CIN, and this really refers to gross chromosomal rearrangements. So here we're using whole chromosome paints that should paint the entirety of these two chromosomes either red and green respectively, but you can see some sort of translocation event has occurred. So what you really have to think about is the changes in gene copy numbers and or their local environments that are associated with both numerical and structural CIN, and how these really help cancers develop. Why do we care about CIN so much? Well, we imagine CIN really to be a nefarious blackjack dealer who's not only shuffling the genome, but bringing in and throwing out cards that really stack the deck in favor of cancer development and progression. CIN is a very important and believed to be an early event in the etiology of cancer. It is certainly a driver of cellular transformation. As I've already alluded to, it's important for tumor evolution or intratumoral heterogeneity. CIN can also drive changes in metastatic gene expression profiles and is associated with the acquisition of drug resistance. So consequently, it's not really surprising that CIN is associated with poor patient outcomes. Now, the current limitations and my research interests are sort of presented on this slide. CIN is certainly highly prevalent in many different cancer types, but the molecular determinants, i.e. the aberrant genes, proteins, and cellular pathways that drive chromosome instability remain largely unknown. So our fundamental question that we try to address through our research is, what are the aberrant genes and pathways that give rise to CIN and contribute to cancer development? Now, some of this work has already been studied extensively in model systems like budding yeast. In fact, a recent study identified 700 out of the 6,000 yeast genes that, when deleted, basically induce a CIN phenotype. And that's about 11.5% of that genome. If those numbers hold true into the human context, we can expect there to be approximately 2,300 CIN genes embedded within the human genome. Unfortunately, however, fewer than 150 CIN genes have actually been identified and validated in human context. So our goal is really to identify a comprehensive list of human CIN genes. Why? Well, simply this will identify the genes and pathways that are normally required to maintain chromosome stability under normal conditions. And secondly, it will give us insight into the aberrant genes and pathways that really drive cancer pathogenesis. And finally, we believe that by identifying those molecular origins of cancer, we can develop therapeutic approaches that exploit those origins for precision oncology benefit. So as I've alluded to already, CIN is very much prevalent in colorectal cancer, and so we often use this as our primary model for study. Just a little bit of statistical information. It is the second leading cause of cancer-related deaths in North America, and this table generally shows you the annual incidence and death rates within the United States in cancer. And you can see that collectively, there are about 175,000 new diagnoses each year, and approximately 63,000 deaths each year as well. Now, unfortunately, in colorectal cancer, approximately 50% of those newly diagnosed individuals are at advanced stages, stage 3 or stage 4. And perhaps even more daunting is that 20% of all new diagnoses are stage 4, with a very low survival rate of about 8 to 14%. So this really highlights to us and many other researchers that we do not have a good handle on how these cancers develop, and therefore we need better understanding of how they develop so that we can tailor better therapeutic strategies to minimize these morbidity and mortality rates. So what I'm going to talk about over the next, the remainder of this particular presentation is our interest in CIN and its relationship with cancer. There's going to be 4 specific vignettes. The first vignette is going to talk about the quantitative imaging microscopy approaches that we developed to be able to look at CIN in a variety of cellular contexts, including cell lines, primary patient samples, as well as tissue microarrays. And so really we're going to show how those assays can be applied to identify the prevalence of chromosome instability or CIN within cancer. We've done this with colorectal cancer, but I'm going to show you application to ovarian cancer. The third vignette is really going to talk about the pathogenic origins. If we can prove that CIN is associated with these cancers, what are the aberrant genes that are actually driving that CIN phenotype? And the fourth one that I'll go into are the therapeutic strategies that are designed to specifically exploit defects in those CIN genes through synthetic lethal paradigms. Some of our work as far as identifying single targets, and more recent work where we're trying to look at combinatorial approaches. So to begin with, I'll start with the first vignette, which is really, can we develop assays to rapidly detect CIN? And this is work that was done by a very talented PhD student, Laura Thompson, with assistance from a summer student with me. And the first assays that we developed are nuclear area analysis. And very simply put, you can imagine that the nucleus is essentially the bag that holds the DNA. So if you stuff more DNA or more chromosomes into this bag, that bag will expand. So shown here on the left are the images. These are the control cells, and what you'll notice for the nuclei in this field of view is that they're all very similar in size and shape. However, when we silence one of our favorite CIN genes, SMC1A, you generally notice 2 differences. First and foremost, these seem to have a much larger nuclear area than those of the corresponding controls. And secondly, we're seeing a lot more heterogeneity. So by collecting series of images from these particular conditions and assessing hundreds to thousands of nuclei, we can get an appreciation for the overall distribution of nuclear areas relative to the various different conditions. So each one of these dots, just to orientate you, represents a single nucleus that was analyzed within these images. And you can generally see with that positive control silencing of SMC1, we again see an increase in the overall range, and of course an increase in the means that is statistically different from those of the untreated or control silence conditions. The second assay that we use is the micronucleus formation assay. For those of you that are unaware, this is a micronucleus here, which is really defined as an extranuclear body found outside that primary nucleus. And the presence of micronuclei are very much hallmarks of chromosome instability. So similar experimental setup to that on the left, when we silence our favorite CIN gene, SMC1A, we certainly see an increase, about a 6 to 7-fold increase relative to those controls. And that, of course, is statistically significant. The final assay that we've really developed and I'm going to discuss today is chromosome enumeration probes. These are fish-based probes that differentially label the pericentric regions of chromosomes 8, 11, and 17. This karyogram simply verifies that within mitotic spreads, we can see the chromosomes correctly localized to the right chromosomes, as well as those pericentric regions. Now, those SEP probes are applied to really assess interphase cells. And so the top image here is really 3 interphase nuclei that are diploid, at least for these 3 chromosomes. So they harbor the expected number of 2 foci for each of those specific chromosomes that's interrogated. Contrasting on the bottom here, what you see is a larger nucleus, which would suggest there may be increases in chromosome numbers. And indeed, when we go and count the number of foci within this interphase nucleus, we see 4 copies of chromosome 8, 3 of 11, and 3 of 17, which is obviously in excess of the typical diploid state. So what we went on to do then was to develop a metric referred to as a CIN score, which really describes deviations from that diploid state. So all 3 of these nuclei have a CIN score of 0 simply because they have the expected number of 2 copies of each of those chromosomes. However, this bottom nucleus has extra copies of chromosomes. In essence, 2 extra copies of chromosome 8, an extra copy of chromosome 11, and an extra copy of chromosome 17, for a total CIN score in this case of 4. Now, by analyzing a series of images and upwards of hundreds of nuclei from a given condition, we can really get an appreciation for the deviation that is observed in these various different conditions. So having developed these particular assays, we then wanted to apply them and have applied them in a variety of different cellular contexts. And what I'm going to present to you in the second vignette is really our efforts to discern whether or not CIN is associated in dynamic in epithelial ovarian cancer. Now, ovarian cancer is very much known to have genome instability, but chromosome instability really had not been assessed. And this collaborative work done in collaboration with Drs. Alon Altman and Mark Noctillo from the University of Manitoba was driven primarily by Signe and is being extended by Claire and Ali Farrell. So the 3 fundamental research questions we had were, can we assess CIN in EOC, i.e. in those patient samples? Obviously we can, because I wouldn't be here otherwise. Is CIN associated with EOC samples, and is it dynamic with disease progression and or treatment response? So how did we assess CIN in these EOC samples? Well, we used that nuclear area analysis that I just showed you earlier on, as well as the SEP analysis that I showed you earlier on as well. And just to remind you, we're collecting series of images in which we can do that analysis. Now, one of the unique things of this particular study is the accessibility of ascites fluid and tumors that have been isolated from those ascites from some women with high-grade serous ovarian cancers. That allows us unique temporal insight into disease progression and treatment response. So here what happens is the gynecological oncology faculty and residents collect that ascites through paracentesis. We are able to isolate the tumor cells to basically 100% purity. They're put onto microscope slides, the fish-based approach is employed, and then we do the quantitative image analysis for nuclear areas and those CIN scores. And what I'm going to do over the next 3 slides is really just show you 3 specific patients that really provide some fundamental insight into the temporal progression. The first question that we were trying to address is whether or not CIN is associated with epithelial ovarian cancer. Here is a patient who refused any sort of treatment, EOC, so we're really gaining insight into the normal biology of disease progression. In this case, we were fortunate enough to obtain 6 samples that we're able to grow, and those are indicated here on this specific timeline. Now, what I'm going to show you first are nuclear areas. So that y-axis is the change in nuclear area. The x-axis are the different samples in chronological order. And what I first want you to note is that there's a fair amount of heterogeneity in each of these particular samples. That heterogeneity or cell heterogeneity is really an indicator of chromosome instability. And in fact, what this might suggest is as this disease is progressing, the amount of aneuploidy or the level of CIN might be increasing. Similarly, when we look at those CIN scores, again, I remind you that diploid state is set at 0, so anything above that diploid state is showing gains or losses in chromosome numbers. And similar to what you see with the nuclear areas, you can certainly see a fair amount of heterogeneity in each of these samples that's indicative of CIN, and perhaps within that last sample, an indication that perhaps we're seeing it be dynamic and increasing with disease progression. Now, for orientation purposes, I've put a blue dashed line here at a CS score of 8. That indicates that any given sample below that has lost or gained no more than 8 total chromosomes of the 3 that are being assessed. Okay, so the next question is whether or not CIN is dynamic in these particular patients. Here's a different patient, EOC13, who has recurrent disease. We were able to isolate 4 samples, 1 immediately prior to any treatment or surgery, and then 3 in rapid succession after the disease recurred, including sample F, which is corresponding to second-line treatment with gemcitabine. When we look at the nuclear areas, we see again, starting with heterogeneity in sample A, that heterogeneity appears to increase A to C to D, and it's perhaps chopped back a little bit, as shown by the interquartile range, is shown in red, which happens to be the same sample that coincides with that secondary treatment. So this may suggest that this individual's disease is responsive to that particular treatment. Similarly, when we look at CIN scores, we again see that heterogeneity. We perhaps see that increase through samples A through D, and it might decrease a little bit in sample F. So it certainly appears to be dynamic and potentially responsive to treatments in this case. The final case that I'm going to show you is really designed to address the question, is CIN dynamic in resistant disease? So this is someone with resistant disease being clinically defined as disease recurrence within 6 months of the final treatment. We obtained 4 samples from this individual, 1 immediately prior to any treatments, 1 after. She was deemed platinum resistant. We had a 3rd sample and then a 4th sample that was collected while she was undergoing treatment with Calyx, which is liposomal doxorubicin. Similar to what I showed you in the previous one, we see that increasing amount of heterogeneity from B through G, that's chopped back a little bit at H. And we see the same sort of thing with those CIN scores as well. But what I want to draw your attention to here is that same blue line, the CIN score of 8. And what you'll notice in these last few samples is that she has a large number of nuclei with tremendous amount of aneuploidy or CIN within them. So that extensive amount of aneuploidy or high levels of CIN occurred shortly before she ultimately succumbed to the disease. So we think it may be a biomarker indicating worse patient outcome. So what did that extensive CIN actually look like? So this is that same patient. You can see sample B is shown here. The nuclei, there is a little bit of heterogeneity, but not a lot. Contrast that with sample G, which is immediately post being deemed platinum resistant. And you see very dramatic differences. First and foremost, you see a lot more heterogeneity. You see increases in nuclear shapes and sizes. And you see a lot more of those foci within each of those nuclei. All 3 of these images are actually taken to scale, and this is actually a giant nucleus found within the sample. And when we did the counts of those 3 specific sets, we noted that there could be upwards of 43 copies of chromosome 8, up to 33 copies of chromosome 11, and up to 22 copies of chromosome 17. So well in excess of the normal 2 that we should normally find. So as far as the summary and conclusion for this vignette are concerned, we certainly can detect and assess CIN, that it's prevalent and dynamic in EOC, and that it changes with time, treatments, and drug resistance. The data also suggests that CIN may be a novel disease biomarker that may have clinical relevance for progression and/or treatment response. However, we do recognize that we need larger patient cohorts to really flesh that information out. And so we're obviously expanding into additional samples with a particular focus on high-grade serous ovarian cancer, because the majority of these women ultimately succumb to drug-resistant disease. Ideally, we want to expand this to circulating tumor cells. Why? Simply put, only about 30% of women present with ascites, so we think this is a great way to expand our analysis to all of those women potentially in other cancer types as well. So having really demonstrated and shown that CIN is prevalent here in ovarian cancer, we then wanted to go on and discern whether or not we could identify and characterize the CIN genes that are causing that chromosome instability in those various different cancer types. Again, this is work done by Laura Thompson. So here, Laura used a multiplexed approach for high content imaging, where she was assessing both the nuclear areas and the micronucleus formation that I showed you earlier on. And the fundamental question here is, can I identify candidate CIN genes that are capable of converting a karyotypically stable cell line into a karyotypically unstable cell line simply by decreasing their protein expression? So what she does here is take those karyotypically stable cells. We use two different cell lines for this screen. She feeds it into a library, an siRNA-based library that has a variety of different elements to it. It has controls, both negative and transfection controls, as well as positive controls, such as SMC1A that I showed you earlier on. And in this case, it has 164 candidate CIN genes that were identified through cross-species candidate gene approaches. So these 164 genes are the human orthologs of subset of the CIN genes that had been identified in yeast. So those genes are silenced within those karyotypically stable cells for about 4 days, upon which they're fixed. They're counterstained, subjected to the high content imaging, where we do the CIN phenotype and do the quantitative analysis for the nuclear areas and micronucleus formation. That allows us to prioritize those particular genes and do subsequent time-consuming cytogenetic analysis to really validate those CIN genes. So what I'm going to show you now are only the results from one of those screens for the nuclear areas in one of the cell lines. In this case, it's HTERT, which is a karyotypically stable immortalized fibroblast. The negative control is shown down here in red. And what you'll notice is we identified 112 putative CIN genes that induce statistically significant differences in the overall distributions relative to that control. Those in blue basically are statistically significant decreases in nuclear areas, whereas those in green are statistically significant increases. And what I want to draw your attention to is this yellow one here, which is SCIP1, because I'm going to go into greater detail in subsequent slides. We also did this for HT1080 cells. Shown here are the micronuclei data from the screen in those HT1080 cells. These are a karyotypically stable fibrosarcomal cell line. This assay is a little bit different. We're looking for the appearance of these micronuclei and the frequency in them. And so what we can see here in red is the negative control, and anything in green on the right basically exhibits or exceeds the minimum threshold required to be identified as significant and worth pursuing. And that level of significance is 2 times the standard deviation of that negative control. Again, I highlight this yellow one, which is silencing of SCIP1, which certainly also induces increase in micronucleus formation. So collectively, these two assays and those two different cell lines give us this Venn diagram. On the left, you can see the 2 cell lines for nuclear areas. On the right, you can see those same 2 cell lines for micronucleus formation. The surprising thing, perhaps, is that 148 out of 164 of those genes that were screened gave a positive result in at least one of these assays. But Laura really wanted to go on and to validate a subset of these, and obviously she wanted to go after the best ones. So those are the ones that were actually identified as putative candidates from at least 3 out of those 4 conditions. And the 10 genes that she went on to validate are shown here. She went on to do a lot more drilled down biology and confirmed, yes, they do induce changes in chromosome numbers. But what I'm going to focus on for the remaining part of this vignette is SCIP1. SCIP1 is the S-phase kinase-associated protein 1. It is part of the SCF complex, which is an E3 ubiquitin ligase complex that regulates the expression of hundreds of proteins. And in particular, it's very well known to regulate cell cycle regulated proteins such as cyclin E1. And this schematic really shows how that generally functions. You can see the SCF complex here. You can see where SCIP1 is. That normally attracts a particular substrate. It becomes polyubiquitinated, which targets it for proteolytic degradation by that 26S proteasome. However, what we think is happening when we silence SCIP1 is we lose SCIP1 expression. We no longer have a functional SCF complex. Those substrates are no longer polyubiquitinated or degraded, and as a result, those substrates accumulate. And we think that substrate accumulation induces CIN and/or cellular transformation. So if that was true, what you'd expect is to see that SCIP1 is altered in a myriad of different cancer types. And indeed, we went into the Cancer Genome Atlas to look up some of this information. You can see these are the prevalence and frequency of mutations, deletions, and other alterations within a variety of different cancer types. And I'm going to be the first to admit the alteration frequencies are not great in any of these cases. However, when we look at shallow deletions or heterozygous loss, i.e. loss of one functional allele, those frequencies are much greater. In fact, if we look at some of the cancer types that I was just talking about, ovarian cancer, for example, approximately 45% of all ovarian cancers are expected to have the loss of one SCIP1 allele. And annually in the United States and Canada, that would equate out to about 10,500 people. Similarly, colorectal cancer found in a lower frequency, only about 14%, but that equates to about 24,000 individuals in the United States and Canada annually. And when we go on to look at the expression analysis and impact on patient outcomes, we can see that individuals with colorectal cancer that have low SCIP1 mRNA expression fare worse than those with high levels of expression. So collectively, these clinical data shown here in combination with the initial screen results certainly support the possibility that diminished SCIP1 expression may be a pathogenic event in the development of a myriad of different cancers, and in particular colorectal cancer. So Laura went on to do a lot of validation. She went on to demonstrate that she could use individual siRNAs denoted by -1 or -2, or a pooled siRNA-based approach, which is comprised of 4 independent siRNAs. She could very much silence SCIP1 in these hTERT cells, and that silencing corresponds with statistical increases in nuclear areas, as well as micronucleus formation. So certainly supportive of the initial screen events. And of course, she extended this to a myriad of other cancer cell lines as well and saw very similar findings. She then went on to actually look at the chromosomes and look at numerical CIN. So here's the control where we can see 46 chromosomes. When she silences SCIP1, she certainly sees differences that can certainly occur. In this case, we counted only 45 chromosomes, which obviously is a loss of one, so a small scale loss. In this specific example, she sees basically a doubling of the genome to 92 chromosomes, which is obviously a large scale gain. And in fact, through further scrutiny of this particular image, you can actually see that chromosomes appear to have been doubled, so they're paired up in each of these particular cases, and that's suggestive of endoreduplication, which is a subsequent round of S-phase independent of any sort of cytokinetic event. She also went on to look at structural CIN, or evidence of structural CIN, and shown here in this spread, you can see evidence from those arrowheads of DNA double-strand breaks within each of those sister chromatids. She also saw evidence for chromosome shattering, which is believed to be a mechanism giving rise to chromatopsis. And of course, she can quantify all of those data, and in those silence conditions relative to control, she's always seeing statistically significant increases. Now, based on this information and further scrutiny of some of these particular images, you could begin to gain insight into some of the aberrant pathways that may be contributing to CIN and these phenotypes. And those phenotypes include replication stress and DNA damage. So Laura went on to do a quantitative imaging microscopy approach where she immunofluorescently labeled cells for RPA, which is a single-strand binding protein, and really an indicator of replication stress. And you can see in the control condition, we see very low levels of RPA, which is expected. However, in that positive control, cells treated with hydroxyurea, we see a dramatic increase in the abundance of RPA labeling that is very similar in the SCIP1 silence condition. And indeed, you can also see the increased sizes of those nuclei relative to the control. And of course, Laura went on to quantify that, and those increases in RPA labeling are certainly statistically significant. Moving forward, she also did a very similar approach for DNA double-strand breaks. She used gamma H2AX, which is a surrogate indicator of DNA double-strand breaks. And rather than just doing total signal intensity, she looked at the number of foci within these particular cells and classified those either below 5 or above 5, and that's indicated in black. So what you can see in the positive control in the SCIP1 silence condition is an increase in the number of cells that harbor more than 5 gamma H2AX foci, basically a 4 to 5-fold increase relative to those controls. So these observations really allowed Laura to conclude that SCIP1 silencing increases replication stress and DNA damage. So she really wanted to know, well, what could be the mechanism that connects SCIP1 silencing with all of these particular defects? And so through literature searches, she came up with cyclin E1. Cyclin E1 is very interesting for a variety of reasons. First and foremost, it is an established oncogene. Secondly, the cyclin E1 gene is genomically amplified and contributes to the development of 20% of all high-grade serous ovarian cancers. And that overexpression, either in individuals or alternately through animal modeling, is associated with increases in replication stress, DNA double-strand breaks, and CIN, and certainly drives cellular transformation. Many of those phenotypes that I just showed you. And surprisingly, increases in cyclin E1 expression correspond with worse patient outcome. And that's shown here for high-grade serous ovarian cancers, where high levels of cyclin E expression, because it's an oncogene, correspond with worse overall survival. So I already alluded to it earlier on. Cyclin E1 is indeed targeted by the SCF complex, at least within the literature. So what we think is going on is this SCF complex is no longer functional due to the loss of SCIP1 expression. As a result, cyclin E1 does not turn over and it accumulates, and it will have implications for the G1S phase transition or replication, as well as centrosome biology. So Laura really set out to really demonstrate some of those particular issues. But first, she wanted to establish the relationship between diminished SCIP1 expression and the expected increase in cyclin E1 levels. So again, she did her indirect immunofluorescent quantitative imaging microscopy approach, where she labeled cells with antibodies targeting cyclin E1. And in the control, you can see a little bit of labeling that's there. Generally, it's cell cycle regulated, so it's only expected to be in a subset of cells. When she silences cyclin E1, it all but disappears. And when she silences SCIP1, it is dramatically increased in virtually all nuclei. And again, those nuclei are large. She certainly can quantify that at the single cell level. So we see large scale increases that are statistically significant relative to the control. And if that wasn't convincing enough, she of course went on to do a Western blot where she could effectively silence SCIP1, and that silencing corresponded with an increase in abundance of cyclin E1. So if the assumption is reduced SCIP1 expression induces increase in cyclin E1 that cause those same phenotypes, if we co-silence SCIP1 with cyclin E1, we would expect to minimize or phenotypically rescue those aberrant phenotypes. And that's shown here in this initial image where we have a SCIP1 silence condition and we have relatively large nuclei. When we co-silence SCIP1 with cyclin E1, we begin to see smaller nuclei, so a rescue of that phenotype. And so she went on to quantify this in the various different conditions that are shown here. I'm really only going to draw your attention to the SCIP1 silence conditions, that when she then goes on and does the dual silencing, she sees a statistically significant decrease in the overall ranges or interquartile ranges, as well as the mean values. Now, those data presented a slightly different way are shown here. We're looking at the cumulative distribution frequencies of those nuclear areas. So shown in gray and black are the SI control and cyclin E1 silenced conditions. Shown in red are the skip1 silenced condition, and shown in blue is the dual skip1 cyclin E1 silencing condition. And what I want to draw your attention to is that this blue line is moving away from those reds back towards those negative controls, although not all the way. So this certainly identifies that a part of this mechanism involves the accumulation of cyclin E1. And of course, she went on to look at micronucleus formation and aberrant chromosome numbers, and each of these relative to those skip1 silence conditions are basically decreasing these aberrant phenotypes by about 50%. So this really shows that at least a part of this mechanism is going through aberrant cyclin E1 expression. So as far as the summary and conclusion are concerned for this vignette, there certainly is a high degree of evolutionary conservation of CIN genes, about 90% between yeast and humans. That skip1 is certainly a novel CIN gene. Silencing is associated with increases in nuclear areas, micronucleus formation, and increases in chromosome numbers. It's also associated with increases in replication stress and DNA damage, and increases in cyclin E1 abundance. However, we do believe there are likely to be additional proteins involved in the development of some of those aberrant phenotypes. In addition, this certainly shows that aberrant cyclin E1 turnover induces CIN and suggests it may contribute to oncogenesis in a similar manner to the genomic amplification of the cyclin E1 gene that occurs in high-grade serous ovarian cancer. So this is really exciting because it shows that these aren't changes that are occurring at the genome level, but rather are changes that are occurring at the protein level. So we believe aberrantly high levels of cyclin E1 may be a major contributor to the development and progression of high-grade serous ovarian cancers or perhaps other cancer types as well. In addition, these findings certainly suggest that additional SCF complex members may also be CIN genes. And in fact, members of the lab have looked at RBX1, COL1, and some of the FBOX proteins and show that they are CIN genes and that some of the mechanisms are certainly complementary but also distinct as well. And that is ongoing research. So having identified those CIN genes, the next final vignette is really broken into two. And the first part is, can we exploit defects in those CIN genes to develop precision medicine therapies? And so this really begs the question, well, how exactly do you exploit genes that are missing or underexpressed? And this is done through a concept that's well understood in cancer now that certainly was born out of model organisms like yeast, and it's referred to as synthetic lethality. Now, synthetic lethality is defined as the lethal combination of two independently viable mutations. Now, to put that in simpler sort of context or analogy, I have the following schematic. What you can imagine is that we're trying to exploit a cancer-causing mutation, perhaps one of those CIN genes like skip1 I just spoke about. And we're trying to identify a rare interaction that will exploit the defect found in that CIN gene. I think many of you are familiar with Mentos, and then if you drop Mentos into Diet Coke, you have a nice explosion. And that's a very rare event. You can't take other candies or chocolates or other things and drop them into Diet Coke and have that effect. So what essentially we're trying to do is identify those drug targets or those Mentos that will be able to exploit those CIN genes. And if we can do that at the level of a tumor, hopefully we would have a nice explosion as is sort of demonstrated here. So we're going to shift gears and we're now going to talk a little bit about RAD54B. We've been looking at this gene for over a decade now. It is an evolutionarily conserved gene from yeast all the way up into humans. Both the yeast and the human forms encode functions within the DNA double-strand break repair pathway, and more specifically within homologous recombination repair, which I remind you is that error-free repair pathway. RAD54B is mutated in a variety of different cancer types, such as those that are shown here. And what we showed over a decade ago is that siRNA-based silencing or genetic manipulation of RAD54B corresponds with increases in DNA content that also correspond with changes in chromosome numbers. So RAD54B is certainly a chromosome instability gene that we hope to exploit through synthetic lethal approaches. So one of the nice things about RAD54B is it has a yeast ortholog that has been studied for synthetic lethality for a long time, and there's a lot of synthetic lethal interactors that have been identified. So what we wanted to do is query the human orthologs of those synthetic lethal interactors to see if we could identify some of those synthetic lethal interactions, whether or not they're conserved in the human context. So basically we take cells, these are isogenic cells. One is a control, so it's RAD54B proficient, and the other one is RAD54B deficient. We transfect the cells with those candidate targets or those candidate drug targets. We allow them to grow for about 4.5 days, at which point they're fixed, stained, and imaged, and then we can do our analysis to see how many cells remain within each of those wells. So the genes that were screened, there's about 80 of them that are shown here. These were identified again through those cross-species candidate gene approaches. And how this generally works is we take the proficient control cells or the deficient experimental cells, seed them independently into 2 identical libraries, allow them to grow, and then on a well-by-well basis generate a synthetic lethal ratio, which would really identify those instances where there's a dramatic decrease in cellularity within this condition relative to this condition. So here's what a synthetic lethal interaction would look like. Just to orientate yourself, the proficient control cells are on the left, the deficient or knockout cells are shown on the right. These are both silenced with the same siRNA, and at the end point of the experiment, you can certainly notice a dramatic decrease in the number of nuclei or cells that are remaining within the deficient cells. In addition, the arrowheads are highlighting cells that are exhibiting hallmarks associated with cellular cytotoxicity. And actually, if you really scrutinize those images, they are exhibiting hallmarks associated with apoptosis. So this initial screen, out of the 80 genes that were screened, 32 of them gave a putative hit as far as this type of relationship. And Sajesh elected to pursue SOD1 or superoxide dismutase 1 as one of those candidate new drug targets. Well, why did he choose superoxide dismutase 1? This is it here. Basically, what SOD1 does is it dismutates superoxide radicals down into oxygen and hydrogen peroxide, which is further reduced into oxygen and water. Now, hydrogen peroxide and superoxide radicals are reactive oxygen species, and those reactive oxygen species have essential signaling roles within the cell. But what we reasoned is if we prevented this from happening, we'd have an increase in reactive oxygen species, which certainly causes a variety of different types of cellular damage, and most importantly for us, is an increase in DNA double-strand breaks. Those double-strand breaks can only be repaired through 2 pathways, non-homologous end joining or homologous recombination repair. But I remind you that these are RAD54B deficient cells, so this pathway really is non-functional. So it forces these cells to repair through an error-prone pathway that we think will overload the system and ultimately lead to apoptosis. Now, I want to draw your attention here that SOD1's dismutation activity really requires copper as an essential cofactor, and that'll become important in a couple of slides. So Sajesh set out to prove this particular pathway or this working model, whether or not it's true. So first and foremost, he repeated the experiments with both individual siRNAs as well as a pooled approach. This is, again, a similar image. The control cells on the top, the deficient cells on the bottom, silenced with the same siRNA, a dramatic decrease in the number of cells remaining, and again, showing hallmarks associated with cytotoxicity. That's quantified here, where we can actually look at the number of cells remaining. And the control cells are all shown in white. The black cells are the RAD54B deficient cells, and you can see a dramatic decrease in the number of cells. So these data really confirm that RAD54B and SOD1 are synthetic lethal and further identify SOD1 as a novel drug target worthy of further study. And so part of the reason that SOD1 was also selected was the availability of inhibitors. And inhibitors, of course, is in air quotes, and you'll see why in a moment. We use ammonium tetrathiomolybdate, which isn't necessarily a SOD1 inhibitor per se, but is actually a copper chelator. And if you recall earlier on, copper is that essential cofactor for SOD1 activity. 2-MA2 is not a SOD1 inhibitor, but rather a general inducer of ROS. So we reason that these two compounds would phenocopy the siRNA-based silencing result and be more effective within the Rad54B deficient cells relative to the controls. And that indeed is the case. You can see that the red lines, the deficient cells, are hypersensitive relative to the Rad54B proficient cells, and that a therapeutic window does indeed open up. So that really allowed Suggest to go on and really characterize that initial working model. So the first thing we wanted to prove is that treatment with ATTM or 2-MA2 would induce ROS. On the left is the proficient control cells, and the right are the deficient experimental cells, really drawing your attention to the middle column in each of these particular panels. You can see that treatment with ATTM and 2-MA2, as expected, induces ROS within each of these different isogenic cell lines. And of course, that can be quantified as well. The assumption is we had an increase in ROS, and that would lead to an increase in DNA double-strand breaks. So again, Suggest went on to test that with two antibodies that really are surrogate indicators of DNA double-strand breaks, 53BP1 and gamma H2AX. Again, this is a slightly different experiment where those drugs are pulsed initially into these cells, and then they're washed out, and those cells are allowed to recuperate for 36 hours. So as expected in both of these cell lines, we see an increase in labeling for those particular antibodies, indicating an increase in DNA double-strand breaks. Upon washout, we notice at the bottom here that there is a dramatic decrease in those particular levels within the control cells, but that those levels persist or remain within the Rad54B deficient cells, certainly showing that those DNA double-strand breaks are not being adequately or properly repaired in the Rad54B deficient cells. And of course, we can quantify that, and those differences are statistically significant relative to the corresponding control cells. So with that, we certainly are seeing increases in DNA double-strand breaks. The evidence earlier on suggested that it perhaps was through an apoptotic mechanism. So we went on to, again, do quantitative imaging microscopy, this time looking at cleaved caspase 3, which is basically the point at which the intrinsic and extrinsic pathways converge. So with respect to cleaved caspase 3, we used a positive control staurosporin, which certainly increases cleaved caspase 3 in both of those cell lines. But really, the increases in cleaved caspase 3 were restricted to those Rad54B deficient cells and were not really evident within those Rad54B proficient cells. And again, we can quantify that information here. So that really led to the current working model where we can use siRNA-based silencing or alternatively ATTM or 2-MA2, which all induce increases in ROS. Subsequently, we also identified LCS1, which is a bona fide SOD1 inhibitor identified by Harold Varmus's group. All of these induce increases in ROS that correspond with increases in persistence of DNA double-strand breaks, and ultimately cause death through an apoptotic mechanism. Now, these findings for Rad54B certainly are suggestive that other members of homologous recombination repair pathway may also be sensitive to synthetic lethal targeting of SOD1. So we've certainly begun to investigate other genes that encode functions within that pathway and have shown that SOD1 does indeed have broad spectrum applicability. So for us, that's very exciting, and we're going to continue this work in sort of the combinatorial approaches that I'll now show you. So this really leads to the end of this particular vignette. Can we enhance the therapeutic killing that we saw through the use of combinatorial approaches? And this is work done by Aaron and Chloe a couple of years ago. So here we're trying to identify synergistic combinations of drugs. So I didn't show you the data. You're going to have to trust me that Rad54B is synthetic lethal with BMN673, which is a PARP inhibitor. So we're looking at everything in the context of BMN 673 and changing those concentrations in the different drugs for drug 2. So just to draw your attention and make sure you understand the concept, drug 1 by itself has a particular effect, perhaps a certain amount of killing. Drug 2 in this case has an equal amount of killing, such that when you add the two of them together, if it's additive, it's simply the product, the result of these two added together. If it's antagonistic, it's below that additive effect. And if it's synergistic, which from a clinical perspective is what we really want, then it has, if you will, super killing in this particular case. So shown here are just one example of each of those three categories. We have BMN 673 with 5-FU or 5-fluorouridine, which is usually part of a chemotherapeutic regimen for colorectal cancer. You can see the two different drug concentrations shown there. And this axis is basically the percentage of cells remaining. This is an additive result. You can see BMN 673 with 2-methoxyestradiol has an antagonistic effect. So it's not a combination we would want to pursue. And BMN 673 in the context of LCS1 certainly has that synergistic impact or that sweet spot that we would want to use moving forward. Now, from a clinical perspective, obviously, these are the ones that we want to identify and move forward through preclinical tests. But these combinations are also important because you would not want to combine these and give them to patients because these potentially could increase tumor aggressiveness and or growth. So what I'm wanting to show you on the next couple of slides are some of the movies that we've generated to look at these combinatorial approaches. And we're really doing this using that biotech system that we have, which is the BioSpa 8 in combination with the Citation 3. So these experiments are performed in 96 well round bottom plates. Cells are seated in that, and what you can see here initially are those individual cells, and as this movie plays, you'll see them coalesce together, form that tumor spheroid, and begin to grow over the time course of this imaging, which is roughly 2 weeks. And so what this really allows us to do is basically try those various different combinations to see if they impact on cellular growth. And this particular 3D spheroid model is obviously much more akin to what you might see in an individual, although it's very much smaller, and it's more representative than the traditional 2D culture systems that are used by many groups. So this next movie really begins at this time point here. This is where that tumor spheroid has formed, and it's at this point where BMN 673 as well as LCS1 have been supplemented into the media. The media is also supplemented with propydium iodide, which is a fluorescent marker of death. And so as we watch this particular movie, what you'll see is those cells begin to die in that tumor spheroid, beginning from the outside and working their way in towards the core of this particular spheroid. And of course, by doing replicates and multi-wells for all of these various conditions, we can basically use that fluorescence indicator as an indicator of cell death. And so here we can actually quantify that. We have 3 different conditions, BMN 673 by itself, LCS1 by itself, and the 2 in combination. You can see the concentration of BMN 673 that we're using really has no effect on death in these particular tumor spheroids, very low concentration. However, LCS1 does appear to induce death as a result. But when we combine those 2 drugs in those identical combinations, we see this purple line, which is indicative of greater levels of killing, so that synergistic effect. So for us, this is very exciting work that is leading us to additional preclinical models to identify those best combinatorial approaches to migrate forward. So as far as an overall summary of today's talk is concerned, you hopefully can see how we're interested in CIN and its relationship with cancer, how we developed those quantitative imaging microscopy approaches and applied them to identify the prevalence of CIN in cancer, to identify those pathogenic origins or CIN genes, and how we can exploit that information to develop therapeutic strategies that selectively exploit cancer cells harboring defects in those CIN genes, as both single agents as well as through combinatorial approaches. And finally, I'd like to acknowledge those individuals and groups responsible for this work. Certainly I'd like to highlight all past and present lab members, as well as a lot of fruitful collaborations. In particular, I'd like to highlight Brad Larson, who was instrumental in assisting us with our micronucleus formation assays, and certainly we'll be doing a number of presentations later today as well. I'd also like to highlight and thank CancerCare Manitoba, the tumor bank in particular, and all of those women who provided their biospecimens for us. I'd also like to thank all the funding groups that are shown along that bottom row. And finally, a special thank you to Biotech for being a wonderful partner that's not only assisted us to advance our research, but for their invitation to speak to you today. So with that, I thank you very much and would be happy to answer any questions.
Thank you, Dr. McManus, for that informative presentation. We will now start the Q&A portion of this webinar. And if you have any questions you'd like to ask, please do so now. Just simply click on the Ask a Question box located on the far left of your screen. So let's get started. We have quite a few questions already coming in. Dr. McManus, our first question is, can you describe the micronuclei assay in more detail? How is this quantitated? Can it be fully automated?
So thank you for the question. Yes, I'll start with sort of the end of that question. Yes, it can and is actually fully automated. So you essentially collect your images. We generally collect between 9 and 16 images per well in a 96 well plate, and the analysis is basically done by hitting a button and getting the number of micronuclei out at the end. So just a little bit more information on the micronucleus assay itself. How it generally is done is in an automated fashion. It first and foremost creates a mask around the nucleus, the primary nucleus itself. We then set a threshold for the distance from the nucleus, which is basically used as sort of a surrogate indicator as to the cell body. And so that's a secondary mask. And then we ask the program, what sort of bodies or nuclear stained bodies are found outside the primary mask, but within that sort of cell boundary mask. And it can give us the number of micronuclei, quantify the number of nuclei, so we can create that as sort of an overall percentage, and we can see changes in those frequencies depending on the various different conditions, and statistically compare those with relevant controls.
Thank you so much. And is it surprising that we see amplification of SKP1 in some cancers, for example, CCR, and the stomach?
So as you would have seen in some of our slides, we certainly were looking at SKP1 and its relationship to diminished gene expression. And that's what we were particularly interested in, with a specific focus on single allelic loss, heterozygous loss, or sort of those shallow deletions. It's not necessarily surprising in a variety of different cancer types that you see a variety of different things happen for any given gene. The question is to discern between those driver events and those passenger events. So as predicted, if you take your favorite gene, you certainly will see instances where it can be amplified or instances where it can be lost. It's really important to do those functional analysis to discern whether or not those gains or losses are those critical aspects. We by no means suggest that SKP1, loss of SKP1 expression, is the only way that we could get some of those particular phenotypes. And in fact, with some of the genes that we're studying independent of SKP1, we can show, and we're aware that these genes can be diminished in expression or increased in expression, and both contribute to those CIN phenotypes and could be pathogenic events.
Thank you so much. And we have time for one more question. What is the role of chromosome alignment, if any, in the pathways that you've been describing here?
So as far as chromosome alignment, I'm assuming what you mean is actually congression errors to the metaphase plate during mitosis, or alternatively lagging chromosomes as chromosomes segregate. So we certainly can do live cell imaging microscopy and quantify those. There's certainly individuals that do a lot of lineage tracing. We don't tend to do a lot of that with our particular genes that we're interested in at the moment, unless we see evidence for there could be congression or segregation errors in those. We can also capture those events independent of live cell imaging, basically in traditional endpoint fixed. If we survey enough cells across enough cover slips, we certainly can see evidence and have counted and scored congression errors or lagging chromosomes as well.
Dr. Kirk McManus, thank you so much for your time today and for your important research. I also want to thank our audience for their questions and for your participation. Join the rest of our presentations today at this conference. Thank you all. Have a great day. Bye-bye.
My pleasure, and thank you very much as well.